So thank you very much for inviting me to come and speak today. I'm going to speak on a little bit of a different topic as far as looking at molecular diagnostics and how they play a role in rapid diagnostics. And so I'm going to have a little bit of overlap information with the other speakers who have already introduced this topic very well, but hopefully you can see a few new pieces of information from this talk. So please. So as everybody's talked about today, there's different ways of combating bloodstream infections, especially bacteria that have these different resistance markers in them. And so we are a small company at Nanosphere. We're about 140 people. We're based in Chicago. And we are a molecular diagnostics platform. So when we looked for different applications to apply that system, we looked at this area, which is in the bloodstream infections area. So as the different speakers have talked about, there's certainly this area of universal decolonization. There's different screening mechanisms, and we'll talk today about the rapid diagnostics area. Now, we're certainly not the only company in molecular diagnostics, but we are one of the few companies that have different multiplex panels in the area of molecular diagnostics to address bloodstream infections. Next slide. So why do we need rapid diagnostics for bloodstream infections? So here's a couple of points for reducing mortality. So each hour that the administration of appropriate antimicrobial treatment is delayed, a patient's mortality rate increases over 7%. It also reduces costs. So if you can rapidly identify the bacteria and corresponding antimicrobial resistance markers, this reduces the cost of antibiotics and the patient length of stay. And then improved antimicrobial stewardship initiatives come out of rapid diagnostics. So you can minimize patient exposure to unnecessary antibiotics, and you can potentially slow down the spread of antibiotic resistance bacteria, like Dr. Peterson said. Next slide. So looking at different types of bacteria, and we'll talk about this in a minute, there's gram-positive bacteria and gram-negative bacteria, and then other bacteria called yeast and fungi. So a Staph aureus is in the gram-positive bacteria family, and so are Enterococcus and Streptococcus. So when you're looking at these, and specifically looking at Staph aureus, you can look at different clinical and economic outcomes that have been presented already today. But here, certainly when you implement rapid results for Staph aureus blood cultures, you can lead to an average of a 6.2-day reduction in the length of stay and over $21,000 reduction in the cost per Staph aureus infected patient. Rapid methicillin-resistant or MECA reporting for patients with Staph aureus bacteremia results in a 25.4-hour reduction in the time to optimal antimicrobial therapy. And patients with false positive blood culture results triggered by contaminants such as Staph epi, which is a coag-negative staph that Dr. Peterson mentioned, have hospitalization costs much higher than the negative blood culture patients. Next slide. So if you're wondering what happens when these patients who are usually septic and are in the ICU, what kind of laboratory process gets done on their blood cultures, this is the overall process. So the blood is drawn from the patient, and it's put into a blood culture bottle. This is essentially media that the bacteria like growing in. Then it's put on that middle top instrument, and there's all sorts of different kinds of instruments from different vendors, but it actually incubates that bottle, shakes it up, and lets the bacteria grow. And if there's bacteria present, it'll ring positive, and then the laboratorian can take that bottle off and do what's called a gram stain. And this is a microscopic slide stain. And what it does is it looks at the bacteria that is in that positive blood culture bottle, and it can see if it's gram positive or gram negative or maybe gram variable. And they can then phone that result in to the physician, and the physician can take that information and start prescribing some empirical treatment for that patient based just on the gram stain alone. Then the laboratorian will take the positive blood culture bottle and they'll start plating it on different media based on that gram stain. Different media grow different bacteria, so they'll want to know if it's gram negative or gram positive and then plate it. And you can see that there's some colonies here growing on this media, and so they have positive growth. And so they have take this growth and they use a different tests, and this is just showing one of the number of tests that are available. These are phenotypic tests. They contain multiple sugars or different types of reagents, and the bacteria grows or doesn't grow in these different wells, and you can tell by the way it grows what organism that is. So after several days of testing, you can get your identification, and then they also look at different methods for getting your susceptibility and whether or not the bacteria is susceptible to different antibiotics through that laboratory. So when we're looking at rapid diagnostic tests, especially when we're looking at developing them from a molecular standpoint, what makes sense is to shorten that pathway. And so what we chose is that after the gram stain, if you could do a molecular test within 2 hours and get the result, it could save a lot of time in the pathway and get you the results so that the physician can act on that information. So next slide. So what we started with was the gram-positive panel. And this is going to be maybe too small for everybody to look at, but these are multiplex panels. So you're looking at taking that blood culture sample, and you're going to be testing it in one test for all these different analytes. So in this menu for this test, which is FDA cleared, it has the Staph aureus, it has the streptococcus, it has enterococcus, it has not only the MECA gene that you're looking for, but it also has some VAN-A and VAN-B genes which code for vancomycin resistance. So there's certainly other companies that are in this area, and they're either cleared or in development. And the FDA actually has really recognized the value, I think, of these different multiplex panels. And so they've really put together some nice guidances for manufacturers like us to be able to take us through this pathway. As Dr. Peterson said, sometimes you need a lot of samples of these different analytes to be able to prove your analytical verification and validation. But we've been able to discuss that with the FDA and be successful in that area. Next slide. So if you're looking at what kind of instrument these kind of tests run on, it's really a very simple instrument. It's kind of like a little computer that you put on your desk, and you just load up some plastic consumables that contain the reagents, and the reagents are going to be looking for DNA, in this case for these kind of tests, but they could also look for RNA, and you're isolating the bacteria's DNA, you're breaking it up, and then you're going ahead and detecting it. So these kind of tests are very low hands-on kind of tests, so they're designated as moderate complexity instruments, and so you do not need to have a centralized molecular diagnostics lab to be able to run these. So a lot of hospitals that don't have that molecular diagnostics expertise can run different panel diagnostics like this. Next slide. So this is going to be very small for everybody in the audience, so let me just describe the main points of this test. This data. So this was a study that's ongoing, and so but giving a few initial outcomes for a test like this, and this is just a Banner Health, which is a complex of different hospitals out west, and here it's showing multiple hospitals, what kind of turnaround time that they were seeing, and from that slide that we saw from your positive blood culture bottle to your identification, they were averaging about 45 hours for all their different hospitals. Here they were getting the results back in 5 hours from the Verigene test, and so they were saving on average over 40 hours to get their result. Go ahead. So as part of that study too, just talking about when we were previously talking about vancomycin treatment, they were able to take that short turnaround time, be able to call up their infectious disease doctors and be able to switch off of vancomycin and get on to methicillin or related antimicrobials within 9 hours. So they're putting together that kind of program at their hospital. Now we talked a lot about gram-positive organisms, especially Staph aureus and enterococcus, but certainly looking at gram-negative organisms, this is an area that will also be playing out over the next couple of years. And the very nice paper that's in your packet talks about all of these gram-negative antimicrobial markers in much more detail. So a lot of these are abbreviated with CTX or VIM or IMP, but they're all related to certain antimicrobial resistance markers that we're concerned about as we go forward. So next slide. So when we talk about resistance and how it comes across the United States, we also look at surveillance programs across the country or the world. And so globally, we see a lot of these resistance markers start in Europe or even Asia or Saudi Arabia or some of these other areas, and they kind of migrate over toward the U.S. As they go forward. And so by surveilling these different areas, we as manufacturers can predict somewhat which markers might be useful in the next couple of years to be able to put on these panel tests. And so this information is very helpful for us and the CDC information and all the researchers out there giving this information so that we can predict what panels might be important going forward. Next slide. So the gram-negative panel for the Veregene product is in development. It's in clinical trials now. But as you can see, it's going to be another multiplex panel having these gram-negative bacteria. It's going to have 6 resistance markers here that are identified in your paper. But certainly an area that we'll be having discussions with FDA on going forward with this pathway. But certainly will be going forward to be able to help with rapid diagnostics. Next slide. So in conclusion, the rapid diagnostics are really linked to improved patient outcomes, reduced healthcare costs, decreased length of stay, decreased pharmacy costs, decreased healthcare associated infections and readmissions, and improved antimicrobial stewardship. So thank you very much.